Hello, welcome again. Today we are discussing about the roles of the liver and we have the key roles of the liver and the other roles of the liver. Let's check about the first role, about the blood sugar regulation. We'll have number 2, thermoregulation. That is about temperature regulation. Our body temperature should be maintained at 7 degrees Celsius. Anything away from that, our enzymes within our bodies will be affected. In case the temperature will be low, what will happen? Enzymes will be inactive. Then when the temperature is high above normal, what will happen? Our enzymes will be denated. So it should just be maintained at 7 degrees Celsius. Let's speak about the regulation of the excess amino acids. The regulation of the excess amino acids, we term it as deamination. So what's this deamination? This is where we convert the excess amino acids. Of which excess amino acids, how do we get it? When you eat the proteins, they will be converted to what? They will have to be converted to amino acids. That's through the digestion process, where it will be absorbed to the bloodstream. And if they will be in excess, they will have some effect to the body cells. So they have to be deaminated. And how will they be deaminated to? We break them to urea. Because of their toxic nature, we first break them to amino acids. Then amino acids has to be broken down to urea, which is at least less toxic and being eliminated as urine. Then we have now detoxification. Detoxification. We don't have an E here. Detoxification. So what about this detoxification? Detoxification is where we eliminate those toxic wastes from the body. An example here, we'll have some things like ammonia, which is toxic. And again, we can remove what? We remove some hydrogen peroxide. Hydrogen peroxide. Those are the two wastes which are eliminated through this method. So let me take you through the thermoregulation. The blood sugar regulation, I've done it in my other video, so you can just refer. You can type blood sugar regulation. I think I've explained when it's high above normal and when it's low below normal. Let's discuss about thermoregulation. What I will say about thermoregulation is about temperature regulation. Body temperature should be maintained at 37 degrees Celsius. This one is for human body. So what will happen if it's high above normal? When above normal, that is above that 7 degrees Celsius, what will we detect? It will be detected by thermoreceptors. These thermoreceptors will be found still in the hypothalamus. So what will happen? These thermoreceptors will send impulse, which sends impulse to which point? To our liver, to the liver. So what will the liver respond by doing? Liver undergoes. We are saying our what? It's high above normal. We need to reduce our reaction. So it will undergo endothermic reaction. Endothermic reaction. That is our liver undergoing what? Endothermic reaction. When it undergoes endothermic reaction, what will happen? It will be absorbed by the liver from the blood. I get the point. We are saying what happens? The liver undergoes endothermic reaction. We have exothermic reaction and endothermic reaction. But when it's high above normal, we undergo endothermic reaction. And what will happen? It will be absorbed by the liver from the blood. Then we have number 2, we reduce metabolic reaction. Reduce what? The liver will reduce metabolic reactions or reduced metabolism, right? And what will happen at that point now? Body temperature reduces or falls, reduces stroke falling back to normal. And it will be very essential for the betterment of the working of the body temperature. We are saying the body temperature should be maintained at that 7 degrees Celsius, right? So our body temperature can go high beyond 37 or it can go low below that 7. I'm saying when it's high above normal, what happens? It will be detected by the thermoreceptors. This thermoreceptor will send impulse to our liver. Then the liver will do what? It will undergo some endothermic reactions. This one is about absorption. So the heat which will be absorbed by the liver from the blood. You know, one role of the plasma is about heat distribution. So the heat will be distributed within the blood. So within this blood, what will happen? The heat will be absorbed. This will reduce the metabolic reaction or reduce metabolism. And because of that, again, we will reduce metabolic reactions within the liver and the temperature will fall back to normal. Let's discuss when the temperature is what? When the body temperature is low below normal. So when the body temperature is what? Low below normal, what will happen? When it's low below normal, when body temperature is low below normal, still again it will be detected by hypothalamus. It will send impulse to which point? To the liver. What will the liver do? Liver now undergoes a different reaction, undergo exothermic reaction. The liver undergoes what? Exothermic reaction. And again, we'll have what? Increased metabolism to generate more heat. So when this liver undergoes exothermic reaction, heat will be released. Is that clear? So we are speaking about here, heat is what? Released. And once it is released and there is increased metabolism and we generate more heat, the heat is released to, where will it be released to? We release the heat to the blood and it is transported to the rest parts of the body. The rest parts of the body. And the end reason here is, and body temperature will do it. Thus, body temperature rises back to normal. Let's again do another of the same. We are saying when our body temperature is low below normal, what happens? It will be detected by your hypothalamus. Which part? Thermoreceptors. The thermoreceptors are the one which will be detecting in the hypothalamus. When it will detect, it will send your impulse, not that your impulse, but that impulse, sorry, to the liver. What will the liver do? The liver will undergo some exothermic reactions. When this liver undergoes some exothermic reactions, it will be released. Once this heat is released, we also have increased metabolism within our liver. And when we have increased metabolism, we generate more heat. What will happen to this heat? We release the heat to where? We release the heat to the blood. And once in the blood, it will be transported to the rest parts of the body. And we say that heat is distributed by the blood plasma, right? So the rest parts of the body. And the body temperature now will tend to do it, to rise back to normal. It will rise back to normal. It will rise back to normal. Let's discuss about what? Let's discuss again about this other one, which is we discussed about our number 3, and we have said our number 3 is deamination. Deamination. What are we deaminating? We are deaminating excess amino acids. Where do we get the excess amino acids? When someone takes proteins, these proteins are very toxic to the body cells. And because now, when they're broken down through the digestion, those proteins are converted to amino acids. So amino acids will be absorbed to the bloodstream. When they will be in excess, they will be very, very toxic, right? They have to be deaminated. And when they are deaminated, we are deaminating these excess amino acids to what? Ammonia. We are breaking amino acids to ammonia. Let me use a table format, and we have excess amino acids. We are saying we are breaking them down to what? Ammonia. This ammonia is toxic. The ammonia is what? Toxic. The excess amino acids, we are saying they will be deaminated to ammonia, right? And how are they deaminated? They have first to be broken down to excess amino acids. We are speaking about these amino acids. We break them to 2 things here, of which are those. We break them to carboxyl group, and we have amino group. Let's know how we got this ammonia, right? The excess, the carboxyl group will be oxidized. This one will be oxidized to release to glucose and used for respiration, used for respiration. That is the first one. Amino group combines with hydrogen atom. Hydrogen, right? Atom. You get your ammonia. And we are speaking ammonia is what? Toxic. And this is what we are speaking about. So the excess amino acids are deaminated to ammonia. So we are saying before the deamination, those excess amino acids, we have to break them to carboxyl group. We are breaking it down to what? Carboxyl group and amino group. So we are breaking it to 1 and 2. This carboxyl group we oxidize into what? Glucose. And you understand the role of the glucose. What do we use glucose for? We use glucose for respiration. Then second thing is about the amino acids. Amino acids are broken down to hydrogen atom. And the amino group will combine with the hydrogen atom, we get ammonia, which is very toxic. So this toxic material, that is now ammonia, will combine with carbon 4 oxide. And it will enter, so we are saying this one is what? Toxic. It will enter a stage called ornithine circle. And what do we get? You get urea plus water. This one is what? Less toxic. So let's explain what happens. And this one now will be transported to the liver. Not to the liver, it's in the liver, it will be transported. This has to be transported to kidney. We transport to kidney, where it will be eliminated as what? Urine. And the last one is about detoxification now. Detoxification. What about detoxification? Detoxification is about removal of this toxic waste. So this one is elimination of excess, not excess, but toxic waste. Toxic waste to either less toxic or harmless waste. And here we just use examples. You use example, and one of our example is ammonia. We said ammonia is what? Toxic. So we can use our ammonia. What was ammonia doing? We break ammonia. We combine ammonia. It's a combination process. Ammonia plus CO2. What do you get? You get urea plus water. And this one is ornithine circle. And we have our arginase enzyme. We have our arginase enzyme. We have the arginase enzyme. So we are saying we have the arginase enzyme. So we are speaking about detoxification. When we are just speaking about detoxification, it's just about elimination of our toxic waste, to less toxic waste or harmless wastes. Of which this one, you just use an example. And our first example is the one that we obtained through the deamination process. And which was that? Ammonia. So ammonia, we do something called addition. We have reduction and we have the addition. So the addition way, we add, that is a combination method, we add with carbon dioxide. What do we break? We break it to urea, which is what? Our urea will be less toxic. Plus water. You know water is not toxic. So, but it's in the ornithine what? Ornithine circle. And in the presence of the arginase enzyme. Let's use the second example. And our second example will be, we will be using the hydrogen peroxide. Where is hydrogen peroxide produced? It's released by the respiring tissues. Which are the examples of these respiring tissues? The examples of the respiring tissue. We can use an example. In the lab, you'll be using a muscle tissue, or you can use a potato. Let's use the one from a muscle tissue. We are saying the examples, you use a muscle tissue. You can use a liver. You can use a potato tuber. Any of those ones. So let me explain now. What are we breaking? Our toxic waste. What will be our toxic waste? Hydrogen peroxide. What are we breaking this hydrogen peroxide? We are breaking the hydrogen peroxide too. What are we breaking? Hydrogen. So we are speaking about, hydrogen peroxide. We are breaking hydrogen peroxide to water plus oxygen. We should have enzyme here, which is what? Catalase enzyme. So when you break this one, your chemical equation will be H2O2 in the presence of catalase enzyme. You get, you're saying water plus oxygen. We can balance our equation, which will be 2, then 2. I think it's balanced. So this one is a toxic waste, and we break it to harmless waste. So we're speaking about what? Our second example of detoxification, where we were using what? The toxic waste, hydrogen peroxide. And we are saying this hydrogen peroxide, we do what? In the hydrogen peroxide, we are breaking it to water and oxygen. Our hydrogen peroxide, we use the catalase enzyme. So the water and the oxygen are harmless. So we are speaking about detoxification. You either break it to less harmful or harmless. And the one which will break to less harmful will be which one? We are breaking down ammonia to urea plus water. Then the one that we can break it to harmless substance is hydrogen peroxide. And you know the effects of the hydrogen peroxide if it accumulates within our bodies. We have the other roles of the liver, but we will not discuss it today. We will discuss in our next section. And those other roles of the liver are like bile production, We have the other role, which is about the synthesis of some vitamins, like the vitamin K. Vitamin K. We have the other roles, like which are the other roles of the liver. We have said, apart from this other role, we have bile production. We have the synthesis of some vitamins, that is by the plasma protein, like the plasma protein, right? The albumins. We have synthesis of vitamin K, synthesis of vitamin K, and even we have the plasma proteins. We have a protein synthesis, sorry. We have the protein synthesis as another one. Thank you, and you can click the subscription button so that you can get to learn more from my biology lessons as we continue. Thank you for your support as we continue learning. I appreciate it for your audience.